So, before we start, do you guys have any idea as to what enzymes are? Yes? No? Maybe? Well, I'd like you to think about your favorite food. Pizza, ice cream, vegetables, fruits, or any food at all. Do you notice how sometime after you've eaten, you feel the need to defecate or you'll feel hungry again? Well, that's because your body has already broken down what you've just eaten and has already absorbed what it needs from it. And this is done with the help of, you've guessed it, enzymes. So enzymes are biological catalysts that help speed up the reactions of the body, your metabolic reactions. So this includes digestion, liver function, and many other functions inside your body. Okay, so let's begin. What really are enzymes? So enzymes are a kind of protein. In fact, they are the largest and most specialized type of protein. And given that they are protein, they can be made up of a single polypeptide chain or, well, more. And these are secreted by living cells. They're organic compounds with a definite structure, and they are water-soluble globular proteins. Just as I've mentioned before, enzymes are biological catalysts that speed up or slow down the metabolic reactions within our body. And just like inorganic catalysts, they aren't affected by the reactions that they catalyze. Because of this, they can be reused over and over again. And what would happen if we had no enzymes in our body? Well, to put it simply, the metabolic reactions in our body would take so long that life would not be maintained. Okay, so now that we know what enzymes are, let's talk about their characteristics. So first, enzymes are very specific. They're endowed with catalytic power and specificity. So this means that enzymes only work on a very specific substrate or other half. In other words, they're loyal, unlike your ex. Aww. Second, enzyme activity is regulated. So what does this mean? This means to say that enzyme activity can either be stopped or inhibited, and it can also be assisted by other molecules. So the enzymes do so much for our body, and it's important that they're regulated so that they don't break down too much product or they don't make too much product. They just make or break the right amount for our body to work at optimal state. And third, enzymes are involved in the transformation of energy. There are certain enzymes that are simple proteins, meaning they simply consist of amino acids. And there are those that are conjugated, meaning they require a non-protein group called a prosthetic group for their biological activity. Now, what do you call an enzyme with an attitude? Amino acid. Get it? Amino acid. Okay, let's move on. Let's go through the definitions and components of enzymes that we may need to discuss for you to grasp this concept very well. First, we have apoenzymes, which is the term used to describe the protein part of the enzyme. This is catalytically inactive, and they require cofactors to become active. Next, we have cofactors. So cofactors are a non-protein component that some enzymes may require so that they can perform their catalytic functions. This can either be a metal ion or an organic molecule called a coenzyme. So again, coenzymes are non-protein organic molecules that help enzymes perform their catalytic functions. Next, an active site. So this is the specific area of an enzyme where the substrate binds to or attaches to during the reaction. But what is a substrate? So a substrate is the chemical substance upon which the enzyme acts on. And a haloenzyme is a catalytically active enzyme or cofactor complex. So next, an anti-enzyme. So an anti-enzyme is something that inhibits the action of an enzyme on a substrate. And an isoenzyme is an enzyme that occurs in multiple forms within an organism, while a metalloenzyme is an enzyme that, well, from the word metal, requires a metal ion. A prosthetic group. So this is a cofactor that is tightly bound to a protein. Then we have activators. which is a cofactor that is a metal ion. And finally, we have proenzymes, which are also called as zymogens. So this is the term given to describe catalytically inactive enzymes, and these need to be activated before they can perform their catalytic functions. Okay, so now that we've tackled the definitions that we need to know, let's meet DJ enzyme to break down nomenclature. Get it? DJ breakdown? Okay. Anyway, there are 3 ways to name an enzyme. The first way is the uninformative way. The second way is by adding the suffix ase, ase. And the third way is by combining an enzyme's designations. The first way would be the uninformative name. So from the definition itself, the type of name that the enzymes have won't give us a clue as to what type of reactions they catalyze. Examples of such enzymes would be renin, trypsin, and pepsin. So for a lot of you, the second way of naming may be familiar. So this has to do with adding the suffix ase, ase. Now, there are things that you may need to consider, such as the name of the substance or the type of reaction that the enzyme catalyzes. Example would be maltase. So maltase is named maltase because it catalyzes the hydrolysis of maltose to glucose. Another example would be lipase, an enzyme that catalyzes the breakdown of lipids. So G, super G. Let's move on. So the third way of naming is quite similar to the second way of naming, except this time you have to consider both the name of the substrate and the type of reaction that the enzyme catalyzes. Example of this is xanthine oxidase. So xanthine is the substrate and oxidase because it catalyzes the oxidation of xanthine. So xanthine oxidase. So you may have noticed that in naming an enzyme, it is important to be familiar with the type of reaction that the enzyme catalyzes. So with that, let's move on to the classification of enzymes. There are 3 ways to classify enzymes. So the first one is by the Commission of the International Union of Biochemistry, which classifies enzymes into 6 categories. The second one is due to the presence in the cell, which has 2 classifications, and the third is due to specificity, which has 3 classifications. Let's first go over the 6 classifications by the International Union of Biochemistry. So the first of these is oxidoreductases. These are the enzymes that catalyze oxidation and reduction reactions. So the systematic name is based on the donor-acceptor oxidoreductase. So next would be transferase. So these are enzymes that transfer a group from one compound called the donor to another called the acceptor. Next would be a hydrolase. So hydrolases are very common and very simple. They catalyze hydrolysis or hydrolytic reactions. So this is basically when water is added to break down a compound. The fourth type of enzymes are lyases. So lyases catalyze the non-hydrolytic removal of groups, and they usually form double bonds. The fifth type are isomerases. So an isomerase would catalyze the structural changes which would lead to the formation of isomers or epimers. The type of name or the specific names of the enzymes would depend as to what type of isomerism would be present. And the sixth and final type are ligases. So what you have to remember about ligases is that they usually require ATP. And so they catalyze the joining of 2 molecules with the hydrolysis of triphosphate or adenosine triphosphate. Okay, so that was all for the 6 major classifications by the Enzyme Commission of the International Union of Biochemistry. So let's move on to the 2 classifications of enzymes based on their presence in the cell. So the first classification would be constitutive enzymes. So these are enzymes that are always present in the cell. While the second type the inducible enzymes need to be synthesized, and their synthesis is either induced or stimulated by certain metabolites. So they're only synthesized under specific conditions or requirements. So finally, let's classify enzymes into the three kinds of specificity. So first is absolute specificity, where an enzyme only works on a specific substrate. An example is maltase, which only works on maltose. So for absolute specificity, the only one type of substrate would fit into the enzyme's active site, and so the enzyme can only catalyze that specific type of substrate. The second type of specificity is group specificity. So these enzymes are specific to certain types of bonds and the groups that surround it. So these enzymes only act upon molecules that have a certain group, and these groups can be a phosphate group, an amino group, or a methyl group, and etc. So just like the illustration shows, it only acts on specific groups, which are represented by the circle and square substrates. So the third type of enzyme specificity is optical specificity. So these types of enzymes are specific to a certain substrate and their configuration, or their optical configuration. An example would be L-alanine oxidase. So L-alanine oxidase only acts upon L-alanine, or the L configuration of L-alanine, and not its D configuration. So its only substrate is L-alanine. And that's it. So now that we're finished, let's relate the discussion on specificities to these puzzle pieces.
Okay, so these pieces are your enzyme and substrate pieces. So the substrate has a unique shape and chemical properties that allow it to fit precisely into the active site of an enzyme. The active site, like a specific spot in a puzzle, is designed to accommodate the substrate perfectly, just like the correct puzzle piece helps complete the picture. So let's relate this analogy to the three types of enzyme specificity. Absolute specificity means it only fits one specific substrate, like a puzzle piece that fits in only one spot. Group specificity means the enzyme fits a group of similar substrate, so it's like puzzle pieces that can fit into similar but not identical spots. And optical specificity is like distinguishing between mirror image puzzle pieces, where the enzyme recognizes and fits into the right-handed or left-handed version of a substrate. So this is how your enzymes interact in general. They are specific in their reaction catalyze and in their choice of substrates. This is how molecules interact with particular targets in different ways, showing their importance in studying and controlling biological processes. Now, let's test what you've learned from this course. You can pause the video if you need some more time to answer. And that concludes our class for today. But there's still so much more to learn about enzymes. So I'll link a video for those of you who may want to dig deeper into this lesson. And I hope that this is really able to help.